Everyone, can you see the slide? Okay lang, I will proceed. So with your metabolism, actually, so this is the summation of our total chemical reaction. So everything that's happening in our body would be called your metabolism. So we have two types of metabolism or two main processes that we are undergoing in order for us to call, in order for us to know that this is metabolism. So we have two, which is your anabolism and your catabolism. So when we talk about anabolism, this is about building up. It means that instead of breaking up the different biomolecules into small pieces, ang ginihimo taya is that from the biomolecules, you will combine them together in order for you to form this complex compound. So suli ay lang ah. So sa catabolism naman niya, so in your catabolism, this is the breakdown. So it means that from a certain complex biomolecule, break down to siya into monomers. So I think we've learned about that in our prelim. So your metabolism is all about the anabolism plus the catabolism. So how we break down the food that we eat and how do we build them up in order for us to build or to make something out of it, okay? Or to make something complicated out of the food that we chew on, okay? So next, we have what we call uh, the main purpose actually of your metabolism is to use up and to build up your energy. So, your energy, guys, kakita lang kamo ha, basi balaga, duho lang kamo da. So, with your energy, this is, as we all know, this is your adenosine triphosphate. So, in order for us to use the energy, what we should do is to actually undergo your ATP into hydrolysis, into which it will, kita na lang ako niya pointer guys, nga color red. Okay lang, katanan lang. Raise hand
Yes,
biglang katanan
Miss.
lang para basi balaga waka lang dali. Okay. So again, so in order for us to use up all of this energy, we have to undergo the ATP into hydrolysis into which it would be further decomposed into your ADP and your PI. Magsiling ta PI, this is your inorganic phosphate. So this will be your free phosphate group. So this phosphate group, amo na siyang ma-attach naman sa other nga mga ADP mo in order to form again ATP. Tapos doong sinado ma-recycle lang sa saga cells and then that's the time that you will form your chemical gradient and all that stuff that's going to happen inside the cell. So next is the reverse, of course. If you don't want to spend your energy, of course, you would want to build up on the energy. So ang sulit lang sa hydrolysis into which your ADP will be bound again to its phosphate group, and then you will form the ATP. So you have two processes on how to actually build up your ATP. So later lang galian eh. So as you can see in our slide, we have the different energy source, and as we already discussed last time with your lipids is about 9 kcal per gram. So it means that it has the most energy to give in our body. So usually when you eat lipid, mas dugay ka gutomon or mas dugay ka kapoyon. So your carbohydrate and proteins only give you your 4 kcal. So it's more ideal to eat protein and fats, but of course do not forget your carbohydrates since your carbohydrate is still the main process or the main pathway in our body to make the ATP or the energy. So, si protein and fat, be careful because they are harmful to our kidney. So, now, with your carbohydrates, kamo na na bahala na magbasa, but these are the different storage type of our different biomolecules in the body. So, for the body, for carbohydrates, it is the glycogen. For proteins, usually we don't have the storage form of your protein. And in your lipids, this would be our triglycerides. Okay, so you are all familiar with this term. So now let's go to the different energy formation process or how do we make a certain ATP. Okay, so we have two. We have actually the substrate level phosphorylation. Okay, the substrate level phosphorylation. And the other one would be the oxidative phosphorylation. So ano gani guys? Phosphorylation. It means that, anong kakatabo ko may phosphorylation guys? Who can answer? Raise hands, raise hands. Or pauna-una, da bit type sa chat box, be. What is phosphorylation? Sa aton nga mga napang-learn ligad sa enzyme. Miss nalipat na ko tula, miss nalipat ko gani isang lesson ta sa lipid sa ang phosphorylation pa. Okay, so ano, by the word itself, who wants to answer? Ansap, wala ged. Tulog pa kamo da, guys. Okay, so, okay, I'll just answer. So, phosphorylation is actually, of course, you are going to add phosphate to a certain group. Okay, so if you're going to add the phosphate to a certain group, then okay, arakil stated that this is the addition of your phosphate or your additional of your phosphate group to a certain compound or certain element. So in our case, during substrate phosphorylation, usually this is just a conversion of your ADP into ATP. So it's just the adenosine phosphate being donated with a phosphate, and then it will become adenosine triphosphate. Okay? So with that, kita nyo ning succinyl CoA to succinate, phosphoenol pyruvate to pyruvate. These are just example of processes that would undergo substrate level phosphorylation in order for us to give ATP. So in every process, we are just producing one ATP. So the conversion of your succinyl CoA and your phosphoenol pyruvate to its certain compound would just be the production of one ATP unit. So substrate level lang siya because isa lang ang kinahanglan ko nga enzyme or isa lang ang kinahanglan ko nga substrate in order for me to produce one ATP. So on the other hand, we have what we call your oxidative phosphorylation. It is termed as oxidative phosphorylation because literally what we are undergoing here is the oxidation or the redox process. So what would be need what we will be needing in your oxidative phosphorylation won't just be a certain substrate to be converted into a certain compound with a phosphate. Rather, what we are doing here is to have electron carriers or electron carrier compounds such as your NADH, NADH and your FADH2. So these two are actually used in our electron transport chain. So we will understand everything later. So your NADH and FADH2 are electron carriers. So it means that these electrons would be the one that would be facilitating the production of our ATP later in our ETC. So oxidative phosphorylation, because we are going to produce ATP via this electron transport carriers and via undergoing your redox reaction. So si NADH ta kasi FADH2 tayo wa ita siya gintapikan directly with a phosphate. Rather, they will undergo redox in order for us to produce the future ATP. So with this one, your oxidative phosphorylation is more advantageous than that of your substrate phosphorylation. Why is it? Because when you read up, your substrate phosphorylation only produces how many units of ATP? It's only one, right? With your oxidative, sa isa mo pa lang ka NADH, you could produce 3 ATP with it. Okay, so put it on your notes. And with your FADH2, what we are actually producing here would be your 2 ATP. So 1 NADH or NADH would produce 3 ATP. One molecule of your FADH2 would produce 2 ATP. So 3 and 2. So miss nga so you will learn later nga So with that, let's move on. So we actually have what we call the acetyl-CoA source. So pinaka-precursor actually sang almost all of the pathway in our body would be the acetyl-CoA. Mapa-protein ka man, carbohydrate, or lipid, all of you should be converted into acetyl-CoA in order for you to go in to our Krebs cycle or TCA. What would be the significance of your TCA again and your Krebs cycle in our body? Of course, this is the NADH and your FADH2 generator nga reaction. So siya ning main ta nga ga-generate sang aton nga NADH kag FADH2 kay para makaproduce pa kita more energy on your ETC. So simpler terms pa lang ni Danay guys ha. So usually from your carbohydrate, your carbohydrates from the complex one. You're going to chew on that. And then what happens in the stomach? They will be converted into different monomers. So usually the simplest monomer of your carbohydrate or your sugars would be the glucose. And then your glucose will undergo glycolysis in the body. And when we talk about glycolysis, it means that you are breaking down the sugar because lysis, di ba? So by breaking down the sugar or your glucose specifically, you are going to produce your pyruvate. After producing your pyruvate will be converted into acetyl-CoA, and then your acetyl-CoA will be the one entering our Krebs cycle in order for us to produce more of our, again, NADH and your FADH2 in order for us to generate more energy in our body. So, medyo childhood ruined, guys, no, ha? Sa una nila na ganing glycolysis, breakdown of sugar, amon nila siya, amon siya. So, this is just the overview, but we will go through the different enzyme and the different products in each of the process of your glycolysis. So, prepare. So, now we have, or we are going to tackle the proper carbohydrate metabolism. So, dili lang tayo ma-focus. From glycolysis to ETC lang ano yung aton i-catch up for today because it's kind of long. And then we'll have the other topic next time. Okay? So far, raise hand ba kung daw gets man? Raise hands, guys, for participation. Diba? Mo pa malang tong ato nakuha. So, how do we get the energy? Okay? How do we produce an energy? Okay? So, para hindi na ma-confuse later because we are going to use many terms. Okay? So, we'll move on. Next, we have the glucose catabolism. So, guys, overall, we are talking about now the carbohydrate metabolism. So, it's all about catabolism and anabolism of your sugar. So, first, what we are going to talk about is the first part, which is the glucose catabolism, or specifically, these are your three processes, or as you can see in the screen, this would be your glycolysis going to your ETC. Okay? So now, you have to imagine first that you are in a cell. Ara ka sa pinakagamay nga cell mismo. So if ara ka sa pinakagamay nga cell, each unit of your cell or each cell in your body performs glycolysis up to your electron transport chain in order for them to produce energy, in order for us to be heated up, and in order for us to function as human beings, right? So with this, with your carbon metabolism, okay, so this is what happens. You eat, you break down the sugar or the starch that you are eating from the rice via the amylase in your the amylase enzyme in your mouth, and then when it goes into your stomach, it will undergo hydrolysis and other stuff in order for you to break them all up. So these are your different monomers, naging pangkaon. And then these different monomers, or specifically ang glucose lang man ang makasulod kag ang galactose sa aton lawas or sa aton nga lining sa small intestine. And that's the time that it will reach our blood. And pag reach sa aton blood, that's the time nga manuktok na to sila sa taga-cell nga ilaagyan. Okay? So pag kadto na sa glucose aton cell, what happens is that it will undergo again the glycolysis process up to your ETC. So we have the different carbohydrate pathway. Hindi lang guys glycolysis ang aton i-discuss, but also we will have this different pathway also, the three of them. So we'll talk about this after discussing the anabolism first. So we'll talk about glucose to pyruvate first. The others will be discussed later. Okay? So first, kundi ato ka na, halin ka na sa blood mo. May naglutaw na to nga sugar, nanuktok na siya sa cell. Okay, insulin, please help me enter the cell. Amo nang hambal ni glucose. So the cell now lets the glucose in, and what happens in there? So what happens in there is specifically in the cytosol or our cytoplasm, there is glycolysis. And during glycolysis, these are all the process. So I don't know about you guys, but we memorized this first year pa lang siguro because ambot ang trip sa maestro to namon sa una. Sorry. Okay, so with our glycolysis, we have the glucose up to your pyruvate. So no choice, guys. You have to memorize everything. So, funny lang a story, kay syempre sa college may yara ka man ng ado lagaw-lagaw sang friends after inom-inom. So usually, ang amon nga game sa gabi is if you get drunk and stuff, if you're still studying, ginahimo na mag-memorize makahiyapon sang glycolysis. So, no joke, everyone who graduated pharmacy should memorize all of the enzyme and all of the byproducts of your glycolysis. So now, let's discuss everything that is in here. So, damo-damo ni siya actually guys, unod ang akon slide. But what I will do is to give you the overview and the simplest and the panawag sini explanation nga makasum up no sang tanan-tanan nga ari sa slide. Okay, amat-amaton ta lang. So, from glucose to your pyruvate, there are about 10 steps. Okay, there are about 10 steps. So, your glycolysis is actually your Embden-Meyerhof pathway. So, amo na sang another name. Yeah, actually, guys, aral na siya sa notes sang ako niya. That would be your Embden-Meyerhof pathway. So, don't worry. Aral na siya tanan sa slide. So, this is your Embden-Meyerhof pathway. And usually, what we are producing here in your glycolysis would be your 2-pyruvic acid. It means that your glucose, in one molecule of your glucose, we are producing 2 pyruvates. Okay, so take note. In one glucose or one molecule of your glucose, we are always producing 2 pyruvates. Okay, so our step 1 would be the conversion of your glucose to your glucose 6-phosphate. So from your glucose to glucose 6-phosphate, as you can see, we have the enzyme responsible for that would be your hexokinase. Hexose kinase, or you can just shortcut it into hexokinase. Hexokinase. So by the word itself, kinase, it means that you are going to transfer a certain phosphate group to your glucose in order for it to become glucose 6-phosphate. And where will that be, free inorganic phosphate come from? Will come from. It will come from the ATP. So, ang atong glycolysis, it's all about borrowing energy and also, of course, having a profit or a net profit of your ATP. So, it's like business, kapital ko daan ay duwa ka ATP ah. So, after ko kapital sa duwa ko ka ATP, ano na ni siya, profit na ni siya dayon. So, basically, the first time that you're going to accelerate the glycolysis or to initiate, the glycolysis is you have to use first the energy. Gakalingaw pa mga kamuda, guys? Wait, pakamuda. So, listen up. Again, so from glucose to glucose 6-phosphate, ara ho lang tawan nyo lang mismo sa ngalan sang aton enzymes because we are already familiar with the different naming of our enzymes. So since this is kinase, you are going to transfer your phosphate. It will become glucose 6-phosphate. From your glucose 6-phosphate, you will become fructose 6-phosphate. So your glucose and your fructose are actually isomers of each other. I mean stereoisomers or isomers of each other. It means that they both have the same chemical formula but different lang ila configuration. As you look at it, si glucose ya aldehyde, si fructose ta ketone. So what we will do is to just have this phosphoglucose isomerase. So isomerase, it means that you are just going to do something about the configuration in order for you to convert your glucose to your fructose 6-phosphate. Okay? And now, that's the second step. Your third step would actually be the conversion again of your fructose 6-phosphate into your 1,6-bisphosphate. It means that you are going again to add another phosphate into your fructose. So it will become 1,6-bisphosphate. It means nga ang imo phosphate, ara siya sa ika number 1 nga carbon and ika number 6 nga carbon. And again, you borrowed another ATP sa aton lawas. Okay? So nagkapital ka na naman. And with this, with the production of your fructose 1,6-bisphosphate, you are going to have this glyceraldehyde and your dihydroxyacetone. Okay? Nga formation. So you will have the cleavage. It means that this fructose 1,6-phosphate will have two products. You have the GLAP and your DHAP. So you will please look at the different terms na lang karon sa mga lagba nga notes. Okay? So usually this is your glyceraldehyde and your dihydroxyacetone. And if you notice this, guys, ano na siya? Sila ang mga pinaka-simplest na sugar, right? The glyceraldehyde and your DHA. So it means that we are just breaking them down into smaller monomers. So now, what happens? So in our next step, we have the GLAP hydrogenase. So this is the one that is going to give us now the 1,3-bisphosphoglycerate, and the phosphoglycerate kinase na naman, mahimo na naman siya 3-phosphoglycerate and stuff like that. So damo na siya magkalatabo na naman hasta nga makalabuot ka sa pyruvate mo. So ang ginahimo lang namon sa board exam is to just memorize actually ang pinaka-importante is ang pinaka 1 to 5, and then the phosphoenol to pyruvate nga processes. So via your 1,3-bisphosphoglycerate, what happens is actually ang 2-ATP, ang din naman niya, ang ADP taya naghimo naman siya ATP. Kinase siya in a sense nga ang ginhimo iya, ginkuha iya ang phosphate. So guys, magsiling ka gani kinase, it's either you give or you take something. 'Di ba kay kinase, nag-move ka. So you are just transferring your phosphate. So it's up na lang niya sa enzyme kung gusto niya ihatag or kwaon balik. So as you notice in your functioning here sa aton babaw, ATP to ADP taya, it means that we are donating the phosphate. But in here, as you can notice, go 1-3 ka, nag-3 na lang siya, nag-2 na lang siya. So it means that the phosphate is being given back to the ADP in order for us to pay for the ATP that we used up. Okay? So next, from your 3-phosphoglycerate, you will become 2-phosphoglycerate. So mutase siya ang aton gingamit nga enzyme. It's because magsiling ka gani mutase ano? Again, you are mutate, 'di ba? Mutate. You are doing something in order for it to become a new species. So it became 2-phosphoglycerate, and your enolase now will convert it into PEP, your phosphoenol pyruvate. And with your phosphoenol pyruvate, again, it is a pyruvate kinase. You are going again to donate your phosphate in this para makapuo na naman tas ang ATP. So as you notice, nagamit ta duwa ka ATP pero ginbalikan ta man gyapon sang ATP nga production sa aton nga mga processes on the last part of our glycolysis. Okay? So guys, I think may correction ko lang ni siya. Your aerobic and anaerobic glycolysis, usually they happen sa cytoplasm, di na siya gyapon ang duwa. Okay? So wala na siya gyapon difference. So both of them happens in your cytoplasm. So magsiling ka aerobic, you have the presence of your oxygen. Magsiling ka anaerobic, you don't have the presence of your oxygen. So usually guys, if aerobic ang imo process during glycolysis, your product would always be 2 pyruvates. But if it is anaerobic, if you don't have enough oxygen in the body, your product would be your lactate. And or your lactic acid. Lactate or your lactic acid. And I have an example for you. I think you've seen this before sa mga memes or stuff. But usually, you have to imagine, nag-exercise ka todo. So, nag-exercise ka todo. And usually, when you exercise, you are running out of breath. So, na-notice naman, guro, anong kakatabo after exercise, guys? Kasi din pala gym. Ara ba lang wala ka pa ka-warm up and everything? What happens after? Sila ang mga chika, ta? Okay. Elaine, what can you say? During exercise nga wala ka nag-warm up, anong kakatabo sa imo lawas? Lane? Lopez? Wala si Elaine? Sino man si Richmond na lang gani? Rich, what happens in your body if you exercise without warm up, or usually nagdiretso ka lang sa isa ka exercise?
Yes.
Hello. So what happens?
Kung wala ka warm up miss before mag-exercise, like
based on your experience,
mas guro miss, mas mabatsagan mo gid bila ang sakit lamis after exercise kay Why bilamis na stretch danay before ka mag-undergo sang vigorous nga exercise, miss.
Okay, very good. So, as you all notice, tsakto ng hambal, if wala ka gani naka-warm up, or it means that you didn't have enough capacity sa imo lungs, kaya ang purpose ng aton warm up is to heat up the body and to also practice our lungs into accommodating more of your oxygen during that movement. So, di ba mas extra sakit ang isa ka certain nga parts ng aton lawas if we do not warm up or sobra ta ka-exercise, gusto lakit ang aton lawas. The reason for that is the lactic acid formation. It means that your body is now undergoing the anaerobic glycolysis. Kay tungod nga jutay na lang ang oxygen nga gasulod sa imo. More of pagwa ka na, kay ga-exhale ka na kaga exhale. So sobra mo kakapoy, you are not retaining oxygen in your body. So now, the glycolysis process becomes anaerobic. So if your body becomes anaerobic, you will produce more of your lactic acid, and this lactic acid will be stored in your skeletal muscles, thus giving us the pain in your muscles or giving us the discomfort after exercising. 'Di ba? Do ka na shock namian di ko na sang namati ko sang amo na nga lesson. So you will appreciate biochem sa mga amo ni nga bagay. So with that, let's move on. Pero kung normal lang tayo nga nagapungko ka lang, wala ka masyado effort yung ginihimo, you're just normally breathing in the oxygen, then you will just produce pyruvate and normally produce your ATPs. So, ang anaerobic nga process, again, amo ni siyang ga-produce ang aton lactate instead of your pyruvate. So, with this, so I hope you would answer this at home. So, usually, of course, in your aerobic, we need oxygen. In your anaerobic, we do not need oxygen. The ATP that we are producing in your glycolysis, it depends. It's actually 6 to 8 ATPs, but it is still not the net. Magsiling ka net, hindi pa ni siya ang final tanga product. Because as you can see, we are still diminishing it with the capital ATP that we used up on the first part of our glycolysis. Sa anaerobic na naman niya, duwa lang ka ATP ga ka-produce ta. Okay? The product for your aerobic, again, is your pyruvate. And in your anaerobic, that would be your lactate or your lactic acid. The one producing, para hindi ka mo malipat sa ulihi, amo ni siyang gakos ang aton nga mga muscle pain during exercise. Okay? So, these are just all the terms na gin-use na to sa glycolysis in order for you to study well again. Okay? So, kaya nyo na. And with this, we have actually the summary for the aerobic glycolysis. We have 10 total reactions overall from the hexokinase going to your pyruvate kinase. Reversible reaction would be 7 steps. Okay. Irreversible reaction would be 3 steps. That would be, put it on your notes or meron na di actually notes, guys. So with your aerobic glycolysis, the irreversible reaction would be your step 1, step 3, and your step 10. So again, the irreversible reactions of your aerobic glycolysis would be your step 1, 3, and 10. Miss nga irreversible. When we talk about irreversible reaction, it means that your pyruvate cannot be interchanged into phosphoenol pyruvate via, again, the pyruvate kinase. So, magsiling ka gani irreversible reaction, you would need another enzyme in order for you to go back to a certain process. So, for example, sa step 3, this is one of your irreversible reaction. Your fructose 1,6-bisphosphate cannot be interchangeably converted into fructose 6-phosphate via your phosphofructokinase. What we'll be needing is a different enzyme in order for us to convert this back into your fructose 6-phosphate. So far, gets lang? Raise hands, guys, be, if na-gets. So far? Akong pointer, guys. Dali lang, gid. Dunadula na nga. Okay. Diba? So, we'll proceed. Okay lang man, guys, nga amo ning view ta. Amo na niya. Kaya para hapos na lang ang pointer-pointer. Nabudlayan ko bisang presenter mode kay nagagwa-gwa. Okay? So far okay lang. Naamulan ni aton nga ano. Okay. So with this, again, irreversible reaction would be step 1, 3, and 10 because We don't this different enzymes present on those reactions cannot be used again to convert back a certain product. So si glucose 6-phosphate mo, hindi na siya ma-convert liwat ni hexokinase into glucose. Okay? So we'll be needing another enzyme para ma-convert them back. So the rest are just reversible. It means that, for example, in your step number 7, si 1,3-bis phosphoglycerate kuya, pwede ko siya magamit gyapon ng kinase enzyme, the phosphoglycerate enzyme, into producing back again my 3-phosphoglycerate. So either way, pwede lang kuya ka move back and forth sa akong pathway. And next, we have your rate limiting step. It means magsiling ka guys, rate limiting step. This is the one that will determine on how fast you are going to produce pyruvate from your glucose or how fast are you going to make this reaction work. So, ang ato niya rate limiting step would be the step number 3. Sulat na kamo da guys. So, in our glycolysis, the rate limiting step or the one that will determine the reaction or the speed of your reaction would be your step number 3. That would be the conversion of your fructose 6-phosphate to that of your fructose 1,6-bis phosphate. What happens in here? So actually, rate limiting step siya because your phosphofructokinase, again, your phosphofructokinase practices what we call your allosteric inhibition or our feedback inhibition. So again, sa mga wide mayo kabate, the step 3, which is your fructose 6-phosphate going to your fructose 1,6-phosphate would be the, or is facilitated by phosphofructokinase enzyme, and this enzyme undergoes allosteric inhibition or what we call your feedback inhibition sa aton enzymes. Diba? Kung ma-review ta sa aton enzyme. So magsiling ka feedback, it means that if nakita ni phosphofructokinase na ay yung mga different to nga cells sa aton lawas, they are still producing more of our ATP. Or si Krebs cycle, balaga produce siya damo-damo nga citrate. So it means that your phosphofructokinase is kinda sensitive to the amount of ATP and citrate in the body. Kung makita iya nga si citrate kag si ATP taas sa aton lawas, what will it do? It will say na, ay, at hagan-haganon talangan na yung pag-convert sang fructose 6-phosphate to fructose 1,6-phosphate para hindi ta dayon makalabot sa aton nga punta nga process. Kaya para limited lang ang aton production para hindi ta mag-usik sang energy kag para hindi ta mag-overproduce sang hindi man dapat. Gets lang? Raise hand if gets lang amo na nga theory. So, it is rate limiting because it will determine the speed of the reaction via being sensitive to the amount of ATP and citrate produced on the latter part of our metabolism. So, gets lang? Raise hands, Biff. Gets lang amo na part? Okay, very good. Mi, ang kabalas ng tannins mo kay ka hyperactive. Okay, so we'll proceed. Jutay na lang ni guys. So, para matapos na lang ni. So, I will leave to you the different technicalities such as the different names of the enzyme. Ang pinaka-importante is you know the theory of the borrowing of the ATP, ADP, the substrate, and the oxidative phosphorylation. So, wala na ko na may mahimo. Iya kung paano nyo ma-memorize. But somehow, I memorized this via, syempre, kapila mo basahon. Hindi lang ni guys nga kaisa dapat kinabasa, but many times. So, as sa board exam ko, second year to fourth year, gin-memorize di siya namon. So, wala ta kawala sa ini nga bagay. So, next, these are just summary again. As you can see, these are the processes that are irreversible and these are technicalities ng tanan. Okay, so, explain ko naman halos tanan. So, these are just summaries. So, guys, cytoplasm man siya gyapon ang aton nga aerobic or aton nga mapa-aerobic man or anaerobic, cytoplasm man siya. But sometimes may anaerobic man tangaga katabo sa mitochondria, but somehow it's still on your cytoplasm area. So again, oxygen requirement, aerobic, yes. Anaerobic, no. ATP, 6 to 8 ATPs. 2 ATPs on your anaerobic occurs in, para sa to sa location. And product would be your pyruvate, and aerobic, that would be your lactate. So, in your aerobic, your pyruvate becomes acetyl-CoA. In your anaerobic, so unknown. Okay? So, kung ano lang na siya dra na lang na siya asta. So, next, we have these different intermediates into which uh, si pyruvate mo to siya, di ba, ang final product sa glycolysis. If it happens in your yeast or in your bacteria during anaerobic process, then that's the time that you are going to produce your ethanol or your wine. So I don't know about you, but when I was in first year sa General Chemta, we are making wines, right? Kagi ka mo himo wine? Raise hands. Kagi ka mo himo wine? Sa
Wala na dayon,
laboratory?
miss.
Wala
Wala
na
na dayon.
dayon.
Kaya
Tubo,
na
wala man.
COVID.
Pero pwede ka mo ka-experiment actually sa balay. So, para lang niya sa curiosity niya guys, hindi na siya project or anything. But ang ginhimo na naman sa chemistry class would just be the tanga juice bala. Any kind of tang juice or anything that is concentrated with sugar. Tapos butangan mo yeast. And pag butang mo sang yeast after 1 week, your yeast will be converting all of your sugar into your ethanol. So, ang nagtabo da is anaerobic glycolysis or anaerobic respiration. So, amon guro do after 2 weeks na inom na siya namon. May ginbilin pa kami for graduation. Medyo wala na magiinom do na hadlo kami sang contamination kay syempre hindi man niya sterilized ang mga bottle and stuff. So, you can drink it after 2 days. So, maskin sa pitsil ka lang ma-ferment or any kind of suludlan nga mga pep bottles, yun na bala, experiment lang. Bobo yeast ang imo nga tanga juice or any kind of juice. So, lantawon, hubog ka na dayon guys kung wala kagit pang bakal. So, sa 20 pesos mo kag yeast may pang inom ka na. Okay? Tapos, next, pyruvate to acetyl-CoA. So, this is the main process that we are really interested about. So, syempre, si acetyl-CoA mo gini yung pinaka-importante para to produce more of our energy. Si pyruvate naman, again, hambal ko naman this is going to be converted into lactate if sa tao siya gakatabo and amo ni siyang ga-deposit ang lactate sa aton skeletal muscle, thus giving us the lactic acidosis or the muscle cramps. Amo ni siyang gapa sakit sang aton yung mga tulaan. So now, let's move on. So with your pyruvate entering now your Krebs cycle, your pyruvate will be converted into your acetyl-CoA. So now, what happens during the formation of CoA? So your pyruvate will undergo or will be catalyzed by an enzyme called pyruvate dehydrogenase in order for us to produce the 2-acetyl-CoA or your acetyl-CoA. So, kamo na bahala magbasa. But during a Krebs cycle, what we are doing is just to break down your acetyl-CoA or to convert them into different carboxylic form. That's why tricarboxylic siya. Kami ara kasang citrate, cis-aconitase, isocitrate. All of this contains COOH on their compounds. So, si acetyl-CoA ko again, convert ko lang into these different forms. And via converting into these different forms, you are going to produce again the different NADH, FADH that will be sent again to your ETC. So, ara na siya guys. Si acetyl-CoA ta amo ning equivalent ya kung i-break down ta siya sa tanan-tanan amo ning iya products. And these are the different equivalents of your acetyl-CoA. So therefore, may gaka-produce ta kuno nga about 12 ATP sa ini nga sa isa ka pyruvate may gaka-produce ta nga 12 ATP. Okay? Else? Ano yung factor 10? Next. Tawag na sa aton, but try ta lang tapuson. Pagkalabot na siya sang acetyl-CoA, so karon ta naman na dason ta naman na istoryahan ang lipogenesis and your gluconeogenesis. So sa aton na acetyl-CoA, ari sa lauluhon niyo naman guys, mayara na siya sa note sa dalom. Can I keep selling sex for money, officer? So actually, that kind of ano ni panawag, acronym or mnemonics, I mean, this kind of mnemonics is kind of green-minded, but it's the one that sticks during review. So the I can I keep selling sex for money, officer? That would represent the different products made in your Krebs cycle. So, imagine nyo guys ha, halin ako sa glucose, nag-pyruvate na ko, si pyruvate in-convert ko sa acetyl-CoA, si acetyl-CoA nagsulod sa TCA. Your TCA now happens in your mitochondria. So, from the cytoplasm of your cell, move in ta sa mitochondria, and then that's the time that your acetyl-CoA will be converted into these different forms. And during the conversion of this acetyl-CoA to these different forms, you will produce again the NADH, the FADH2, and so on and so forth. Okay? So after mo na-produce ang NADH and FADH2, what happens is that you will move on with your life and go with your electron transport chain. Tatlo gini silang connected. So in electron transport chain, what happens in there is the oxidative phosphorylation. As you notice, from your glycolysis and your TCA, we have reactions such as your substrate level phosphorylation, into which nga may pasahay lang kamusang phosphate in order for you to produce 1, 2 ATP molecules, 3 ATP molecules. But in your ETC, what happens again now here would be the conversion of the FADH and your NADH from your Krebs cycle or from your glycolysis. Medyo tayo man ito nga ka-produce ng NADH. And then, it will go in with your electron transport chain. So do not read everything in here, but during electron transport chain, what happens is that we are jumping through all of these complexes. I do not advise to really memorize the different complexes, but I want you to visualize kung anong gakatabo. So, di ba, halin ka na sa glycolysis, cytoplasm na itong gakatabo. I mean, sa cytoplasm, we have glycolysis happening there. The pyruvate now, converted to acetyl-CoA, enters your Krebs cycle. It means that it goes into your mitochondria. So, sa pinaka-main yung mitochondrial area. What happens now? After nga nakaproduce na ko NADH and FADH2, what happens? Ma-move in ang akon reaction into your ETC, and your ETC happens where? It happens in your inner membrane or sa mga folds ka aton mitochondria, which we call your cristae. Diba? Your cristae. So, mga inner linings na siya sang aton mitochondria, gakatabo ang aton ETC. So, you have to remember, ara siya ho, lagid siya sa mga phospholipids ta area. So, what happens during your ETC? What happens here is all of the NADH again and FADH produced from your glycolysis and your Krebs cycle goes down here. Ang natabo di, si NADH ta sa complex 1 ta, it donates its electron to this complex. Pag donate ang electron sa ini nga complex, this complex will pump out its proton, which is the hydrogen in here. Si FADH ta naman niya, it bypasses the complex 1. It means that it just goes into your complex 2, and then it donates also its electron in here. And then what happens is that pag donate ang electron niya, there will be a transfer of electrons from this complex 1 to via your QH2 or your ubiquinone enzyme. And then what happens again? The electron will just be transferred again into your complex 3. Pagka-transfer na naman sang electron mo, what happens? This complex 3 again will pump out another proton to this is the outer area or the intermembrane space. Okay, so amo ni siyang inner part sa aton nga mitochondrial membrane. So, pag pump out sang proton mo, what happens again? The electron will move in this area again, and then it will be transferred here in your complex 4. So this is where it gets tricky because your complex 4 now is what we call your oxygen complex or your electron acceptor complex. So what's in your fourth complex would actually just be the presence of your oxygen. So si oxygen ma-receive sa sang imo nga electron. After receiving the electron, it will just form water. So with this reaction again, nag-pump out na naman siya proton. And as you noticed, anong purpose nga? May mga proton ta sa aton intermembrane space. So the purpose of producing or for getting out the proton in your intermembrane space, there will be now difference in your chemical gradient. So this part will become quite negative, ang dringa part sa dalom. This part will become more negative. So ang matabo sina, since there is imbalance of electrical charges, what happens is that it will power up what we call the ATP synthase. This one, ang aton nga complex 5. This will be again our ATP synthase. It means that this is the part that we are going to make our ATP. So, ang tanan nga positivity di nga gakatabo, kag ang tanan nga negativity di nga gakatabo sa dalom, it generates electricity. And via generating this electricity or electrochemical gradient in our cell, what happens? Si ATP synthase ta malibot ni siya. So, I think may wala ko diagram. But usually your ATP synthase looks like a trompo, balahaw, trompo. So the electrochemical gradient will make the ATP synthase rotate. So samtang ga-rotate ni siya, kita niyo ni ho, si ADP mo mismo gina-donatan ta sang phosphate. So every time nga nag-rotate ni siya ang aton nga ATP synthase or which is our complex 5. So please read up the different details. So pagkita ta nga ga-rotate na si complex 5, what happens is that the ADP or our adenosine phosphate binds to an inorganic phosphate in order for us to produce more of our ATP. Okay? So with this, from NADH, FADH, it undergoes oxidation via giving out its electron. By giving out the electrons to the complexes gives out its proton to the intermembrane spaces. And via the transfer of electrons from one complex to another, what happens is that, again, there is still an accumulation of the positive charges, and then the inner cell or the inner membrane of your mitochondria becomes more negative. There will be electrochemical gradient production. What happens is that your ATP synthase or your complex 5 will rotate, and during the rotation of that or during the activation of this, there will be production of your ATP via binding your ADP to your phosphate. So, miss, anong difference dito sa aton nga substrate level? Sa substrate level ya, diretso lang. ADP plus PI is equal to ATP. Sa aton nga oxidative, nag-undergo ko danay sa complex 1 to 4 nga redox reaction para lang makaproduce ang ATP. So far, gets ang difference sa aton ATP production? Raise hands if ka-gets so far sa amo to nga part. So that would be it for our glycolysis going to your ETC. So it's kind of specific when you study it. So these are the different complex 1 to complex 5. So kamo na bahala sang details. But this is the main point. Okay? So from complex 1 to 5, you're just going to transfer the different electrons, proton out, then develop chemical gradient, and then form kasi yung mga. So these are just the summary of how many ATPs that we are producing from the glycolysis going to your TCA. So in order for you to visualize everything that we've talked about today, just look at this table. So as you can see, from glycolysis mo again, summarize na tanan kung diin asta or pila ka glucose ang aton or pila ka ATP ang aton gakahimo sa aton glucose. So with this, kita nyo guys, 2 NADH, 2 NADH akong ga-produce, 3 NADH man de, 1 NADH, pero nga atag 34 kabilog. Because again, when you internalize later on, 1 molecule again of your NADH is equal to 3 ATP, and 1 molecule of your FADH2 would be equal to 2 ATP. Okay? So, kamo guys ang aton yung mga glycolysis to your ETC, but that's the whole thing that you need to study. Okay? So, memorize the enzymes, memorize the step-by-step, the rate limiting, what are the irreversible parts, because we are still going to use it for your clinical correlations. So far, dira na lang taanay because we are at 53 minutes na. So, it's kind of long na. So, okay na? Okay na taanay? Raise hands lang if gets ang lesson. If hindi, just rewatch the video lang. So, wala na problema? Raise hands na. Gwa na ta? Gets na? Gets na? Okay. So, hopefully, you got the lesson and you got the gist kung nga ginahimo taning glycolysis up to your ETC. Okay? And that you could visualize nga halin ka sa cytoplasm, move in ka sa mitochondria, and then via mitochondria, you're going to produce. So, this is where it all makes sense. That your mitochondria is the powerhouse of the cell. So, ara na kamo sa level, guys, nga maghambal ka mo powerhouse of the cell, hindi lang ang panumdumon mo nga, may nag-produce. Utok mo na, gakatabo na ang substrate level phosphorylation, may oxidative phosphorylation na dagiling gakatabo sa membrane amo na nga powerhouse siya of the cell. Okay, so now it makes sense nga mitochondria is the powerhouse of our cell. So that would be our conclusion for today. We'll continue next meeting. And lapit na lang sila klase. Adya, bye-bye. Thank you guys.